Artificial intelligence has come a long way, from Alan Turing's groundbreaking ideas on simple machine intelligence to the advanced AI tools we rely on today. Once a concept from science fiction, AI is now transforming how we live, work, and make sense of the world. Hey guys, welcome back to another video. Today marks the beginning of a special 3-part series on AI and its transformative role in scientific research and bioinformatics. I originally thought about just making one video on this topic, but after organising everything, I realised it would probably run well over an hour and I didn't want the important practical information at the end to get lost. So I've decided instead to split it into 3 videos. In this first video, we're going to explore what's actually happening right now with AI in bioinformatics and genomic research. We'll be covering the AI tools that are readily available for every bioinformatician and scientist like you and me. We'll cover the fascinating ways researchers are currently using AI for scientific discovery, and then also 2 of the biggest challenges we're facing with AI and genomic research today. Then in the second video, we'll take a first principles approach to envision where this field might be heading in the next 20 years or so, especially considering how incredibly fast AI technology is advancing. And then finally, in the last video of this 3-part series, we'll explore the history of technological revolutions, examining both the farming revolution of the 18th century and the more recent internet revolution to understand who thrived and who got left behind during these massive shifts. I'll use these historical lessons to offer some practical advice on navigating your career and how to position yourself for success in this transformative AI era. So with that, let's get started with part 1. How is AI being used in bioinformatics and genomic research today? I'm going to break this question down into 2 parts. First, how is AI helping the everyday scientist or bioinformatician? And second is how it's being used in research to advance scientific discovery. So let's start with how AI is transforming the day-to-day life of scientists and bioinformaticians. Specifically, what AI tools we have that make us faster, more efficient, and maybe even a little less stressed. So here's what the current landscape looks like. We have chatbots that serve as your everyday assistants, tools designed for coding, platforms built just for writing, and AI solutions tailored for scientific research. These AI solutions help with practically everything. They can write code, break down complex ideas, summarize dense research papers, and help you draft anything from a quick email to a detailed grant application or manuscript. I'm sure you're already familiar with many of these tools, so I won't elaborate further in this video, but I can't overstate their transformative impact. When used effectively, AI can make an individual 10 times more productive. Now, I should also acknowledge that they're not perfect, and there are risks with using AI, like hallucinating incorrect information, as well as legitimate concerns around data privacy and ethics. However, these tools and similar ones are simply changing the game in terms of individual productivity and output. Let's now move on and look more specifically at how AI is driving breakthrough research in science itself. When people hear AI today, they're probably thinking about generative AI. This is the technology behind those chatbots and coding tools that we just talked about. But artificial intelligence is actually a much broader umbrella term. It includes foundational methods like machine learning and deep learning, as well as other branches like natural language processing, expert systems, computer vision, and more. These categories often overlap in real-world applications, but in this video we'll explore them as separate entities to showcase the main methods driving bioinformatics and genomics research. Think of it less like a strict hierarchy and more like a landscape of interconnected tools, each with its own strengths and often used together to solve complex problems. So let's start with machine learning. Machine learning is the foundation of AI. It allows machines to learn patterns from data and is divided into different approaches like supervised, unsupervised, semi-supervised, and reinforcement learning. Deep learning, a specialized subset of machine learning, takes this further by using multi-layered neural networks to uncover complex patterns in large datasets. Let's go through some specifics, starting with supervised learning. Supervised learning trains models using labeled datasets, where each example comes with an answer or label. The algorithms used in supervised learning vary in complexity and are chosen based on the nature of the data and the problem. Here are some common models you'll encounter. Statistical approaches like linear regression help predict continuous values such as gene expression levels, while logistic regression works great for yes-no classifications like determining if a gene relates to a disease. Decision trees give us easy-to-follow models that branch data based on clear decision points. When we combine multiple trees, we get random forests, which offer more accuracy and stability. Support vector machines excel when we need to separate data into distinct groups, especially with high-dimensional datasets. Meanwhile, k-nearest neighbors simply classify points based on what's closest to them. For more complex problems, gradient boosting combines multiple simpler models sequentially, with each one focusing on correcting the errors made by the previous model. And for smaller datasets, 𝗕𝗏𝗽𝗍 uses simple probability rules to estimate how likely different characteristics or features belong to each category. Let's look at a real-world example of supervised machine learning in bioinformatics, specifically the support vector machine. At its core, a support vector machine finds the optimal boundary to separate different groups of data. When data is too complex to separate it directly, it transforms it into a form where separation becomes possible. Support vector machines are often used to analyze gene expression data from microarray experiments. If you're not familiar with how microarrays work, here's a quick overview. Let's say researchers want to study breast cancer and compare gene expression between diseased and healthy tissue from the same individual. They might take a tumor sample from cancerous tissue and a control sample from healthy tissue, ideally from the same organ to keep things consistent. The RNA is extracted from both samples and converted into labeled cDNA. The cancerous tissue gets labeled with a red fluorescent dye and healthy with the green. This labeled cDNA is applied to a microarray chip, which is essentially a small surface like a glass slide with thousands of tiny spots. Each spot represents a specific gene, and when the labeled cDNA binds to these spots, the chip is scanned to measure the fluorescence levels. The data shows which genes are active in the healthy sample, the disease sample, and both. Now, support vector machines come into play here to analyze this data. They learn patterns of gene expression associated with disease and can then classify new samples as either cancerous or non-cancerous. So we basically pinpoint which genes are the most associated with the disease to predict whether new samples have the disease, which can inform if and how they need treating. And there's this tool called Gene Pattern, developed by the Broad Institute, which combines visualization of microarray data with support vector machine-based analysis, making the whole process more seamless. Okay, let's now move on to unsupervised learning. Unsupervised learning is a bit different from supervised learning in the sense there are no labels or predefined answers in the dataset. Instead, the algorithm tries to find hidden structures or patterns in the data on its own. This can be really useful for exploring data when you don't already know what you're looking for. There are a couple of common approaches in unsupervised learning. One is clustering. So this groups similar data points together based on their features. Another is dimensionality reduction, which simplifies large datasets by reducing the number of variables while preserving the most important information. So just to run through these, k-means clustering and hierarchical clustering both organize data into groups, but in different ways. So k-means clustering creates a fixed number of clusters, while hierarchical clustering builds a nested structure showing relationships at multiple levels of detail. GMM is particularly useful when your data might contain overlapping groups, while dbScan excels at finding irregular shaped clusters and identifying outliers that don't fit in any group. For dimensionality reduction, PCA offers a straightforward approach for compressing data while preserving variance, while 𝗕𝗏𝗍 is especially powerful for visualization tasks. Autoencoders use neural networks to learn compressed representations automatically, making them suitable for very complex data patterns. Let's look at a real-world example of unsupervised machine learning in bioinformatics, specifically k-means clustering. K-means clustering can be used to group similar samples based on patterns in data. So let's say researchers are studying gene expression across different cancer types, like breast, lung, and colon cancers. For each patient, they measure the activity of thousands of genes using RNA sequencing. This gives them a big dataset where each sample represents one patient, and each feature is a gene's activity level. To make sense of this, they use k-means clustering. The algorithm groups samples or patients with similar gene expression profiles into clusters. Each cluster might represent a specific cancer type, like all the breast cancer samples grouping together, or it might uncover new subtypes researchers weren't aware of before. This is really useful because it helps uncover patterns in the data that can classify the cancer types and track how they progress, and it can even identify new biomarkers for diagnosis or treatment. Researchers often use tools like the Python library scikit-learn and 𝗘𝗏𝗽𝗄 to perform this type of analysis. Okay, another category of machine learning is semi-supervised learning. Semi-supervised learning strikes a balance between supervised and unsupervised learning. It works with datasets that are partly labeled, where only some of the examples come with the labels, while the rest remain unlabeled. This is valuable where labeled data is scarce or expensive and time-consuming to generate. Here are some commonly used models in semi-supervised learning. Self-training and co-training bootstrap from limited labeled data, with co-training adding power by leveraging multiple perspectives. Graph-based methods excel in situations where data naturally exhibit similarity, such as genes with related functions or proteins with comparable structures. The idea with these is simple but effective. By using a small amount of labeled data, the model can start to understand patterns in the data and then apply what it learns to estimate labels for the unlabeled data. And this way, the model effectively trains itself on both labeled and unlabeled data, which greatly enhances its predictive power. These methods have demonstrated the ability to enhance predictions, such as predicting cancer reoccurrence and identifying regulatory sequences in the genome. Okay, let's now go on to reinforcement learning. This is another category of machine learning, but unlike the other approaches, this focuses on training an agent to make decisions by interacting with its environment. The agent learns through a system of rewards and penalties. So when it takes a good action, it gets rewarded, and for a bad action, it's penalized. And over time, the agent refines its strategy to maximize rewards, mimicking how humans or animals learn through trial and error. Here are some commonly used reinforcement learning methods. Q-learning and 𝗘𝗲𝗄𝗏 are core algorithms for learning the best actions. Q-learning always seeks the optimal choice,
while 𝗘𝗲𝗄𝗏 adjusts
based on the path the agent actually takes, making it more cautious. Deep Q networks bring the power of deep learning into the picture, allowing agents to handle much larger and more complex environments that would be impossible with simple tables. They're widely used in real-world applications like video games and robotics. Policy gradient methods take a different approach by directly improving the agent's overall decision-making strategy or policy, rather than estimating the value of specific actions. This makes them particularly effective for tasks involving continuous actions, such as adjusting the steering angle of a self-driving car. Proximal policy optimization (PPO) is a refinement of this method, designed to carefully balance trying new actions and sticking to what works, ensuring the agent learns reliably without making abrupt changes. This is often used in cutting-edge reinforcement learning applications. And then Monte Carlo methods, by contrast, are less focused on immediate reactions and more on learning from the full trajectory of an episode, which is ideal when rewards are delayed or spread over time. Let's look at an example of how reinforcement learning is applied in bioinformatics. So in drug discovery, researchers often want to improve molecules so they work better, for example, making them bind more strongly to a target or reducing their toxicity. Deep Q networks can help by learning how to tweak molecules step by step. The reinforcement agent starts with a base molecule and makes small changes like adding or removing atoms or chemical groups. And then after each change, the molecule is tested virtually to see how good it is. Does it bind better? Is it more soluble, less toxic? The agent gets a reward if the change helps, and a penalty if it makes things worse. Over time, the DQN figures out which changes tend to improve the molecule and learns how to build better drug candidates without needing to physically make and test each one in a lab. A specific framework called MolDQN has successfully used these DQ networks to optimize molecular structures for properties like drug likeness and similarity to known compounds. And tools like DeepChem, 𝗘𝗲𝗏𝗏 and OpenAI Gym provide the building blocks to develop and train these models in real-world drug discovery pipelines. Right, now lastly in the machine learning category, we have deep learning. So deep learning, or DL for short, is a really fascinating area within machine learning that utilizes multi-layered neural networks. It does an incredible job of analyzing and learning from large, complex datasets, which is why it powers so many of the modern advancements in AI. One of the standout features of deep learning is its ability to automatically identify intricate patterns in data. This makes it especially useful for various tasks like image analysis, language processing, and even managing vast amounts of numerical data. Here are some commonly used models and methods in deep learning. Convolutional neural networks are excellent for image analysis and are commonly used in medical imaging like tumor detection and everyday applications like facial recognition in smartphones. They work by breaking down images into smaller patterns and analyzing them layer by layer, effectively recognizing shapes, edges, and other features. Recurrent neural networks are specifically designed to handle data with a sequence or order, like sentences or time series data. They remember previous inputs, making them powerful for tasks where context matters, such as analyzing gene sequences or predicting trends over time. Transformers excel at processing long sequences, analyzing all parts of the data at once rather than step by step. This allows them to be highly effective for language tasks like conversational AI and solving complex problems like protein folding. Autoencoders work by compressing data into a simpler form called encoding and then reconstructing it back into its original state. By focusing on the most important patterns in the data, they can identify outliers or anomalies, which makes them particularly useful for detecting rare events such as mutations. Generative adversarial networks, or GANs, consist of two networks, a generator that creates data and then a discriminator that evaluates it. This competition improves the generated data's quality, making GANs valuable for creating realistic simulations and artistic applications such as AI-generated images or deepfake content. And then self-supervised learning allows models to learn patterns from unlabeled data by creating their own pseudo-labels. For example, a model might predict missing words in a sentence or fill in hidden parts of an image, enabling it to learn from large amounts of raw, unlabeled data. Now, deep learning often works hand in hand with reinforcement learning to improve decision-making. For example, methods like deep Q networks use deep neural networks to help agents handle complex environments and data, such as molecular designs or protein structures. This combination of deep learning and reinforcement learning allows agents to learn from rich, unstructured data, making the learning process more effective. Let's look at one example of deep learning in bioinformatics. So one groundbreaking application you might have heard of is the prediction of protein structures. Researchers use transformer models, which excel at analyzing sequences like amino acid chains and proteins, to predict their 3D shapes. For instance, AlphaFold, developed by DeepMind, leverages transformers to make highly accurate predictions. Understanding these protein structures is crucial for studying how proteins function and for designing targeted drugs. Tools like TensorFlow, PyTorch, which are both Python libraries, and also DeepChem make it easier to build deep learning models for bioinformatics applications. Now, it's worth pointing out that these different areas of machine learning, so supervised, unsupervised, semi-supervised, and reinforcement learning, aren't isolated. They actually overlap quite a bit. Take deep learning models, for example. They can be trained in a supervised way to handle specific tasks, or they can apply unsupervised or semi-supervised methods to identify patterns in unlabeled data. Deep learning is reshaping areas like genomics and drug discovery, and is a hot topic right now, and one that I imagine will expand even more in the coming years. Potentially worth looking into if you're exploring further opportunities. One key challenge though with deep learning in particular is understanding why models make certain predictions. These models are often seen as a black box, as their inner workings can be so complex that it's hard to pinpoint exactly how they reached a decision. This is a huge focus of ongoing research, especially in critical areas like healthcare, where we need to understand why a model predicts a patient has a certain disease. And tools such as 𝗕𝗵, which stands for Shapely Additive Explanations, this is a Python package, and also LIME, which stands for Local Interpretable Model Agnostic Explanations. These are both starting to shed light on these opaque systems, giving us a glimpse into how models make decisions. Okay, so that's it for the machine learning category. Let's now go on to some other categories in the AI landscape. So starting with expert systems. Expert systems are one of the classic branches of AI. They don't learn patterns from data. Instead, they rely on a set of clearly predefined if-then rules that capture expert knowledge to make decisions. One example is 𝗏𝗏. This is a database that uses rule-based pattern matching to identify protein families, domains, and functional motifs. So experts will go in and set some specific patterns or signatures representing known protein characteristics. Then you basically encode these signatures into the 𝗏𝗏 database, and this allows users to scan protein sequences and automatically annotate patterns and features. Right, next we have natural language processing, NLP. NLP is all about teaching machines how to understand, interpret, and even generate human language in a meaningful way. It's the backbone of technologies like chatbots, automatic translations, and tools that extract insights from massive amounts of text. Essentially, it bridges the gap between how we communicate and how machines process information. So let's take the example of literature mining in scientific research. Tools powered by NLP can automatically sift through thousands of papers, flagging key insights or highlighting new trends. You can imagine how much time this saves. Instead of spending weeks combing through articles manually, you can just use these AI tools to fast-track the learning and information gathering process. Okay, next one, evolutionary computation. This is all about solving optimization problems by mimicking the processes seen in natural evolution. It doesn't think or learn like other forms of AI, but instead it works by iteratively improving solutions. The process involves mechanisms like mutation, which introduces changes to solutions, crossover, which combines parts of different solutions, and then selection, which chooses the best performing solutions. Over multiple generations, the algorithm evolves solutions that are closer to the desired outcome. So one practical example is EvoDesign, which is a tool used for designing proteins from scratch. EvoDesign uses evolutionary algorithms to optimize protein sequences for specific structures or functions. The process starts with a random pool of protein sequences, which you can think of as the first generation, and then these sequences are evaluated based on how well they meet certain criteria like stability or function. This is called the fitness score. And the best performing sequences are then selected, combined, and slightly altered to produce a new generation of proteins. This process is repeated over many generations until highly optimized proteins are designed. Okay, next, computer vision. Computer vision is an area of AI focused on enabling machines to interpret and analyze visual data like images and videos. It powers a wide range of applications like image classification, object detection, and medical imaging. One practical application of computer vision in bioinformatics is Cell Profiler. This is a tool used for analyzing microscopy images. Researchers rely on it to measure features like cell size, shape, and texture, helping them to uncover patterns in how cells behave under different conditions, like looking at how cancer cells respond to a treatment. And you can combine computer vision with deep learning models like convolutional neural networks to track changes in cellular activity in real time, such as tracking how cancer cells migrate to better understand metastasis. These algorithms will essentially analyze live microscopy images frame by frame, linking positions across frames to track movement patterns. And another powerful example of computer vision is in breast cancer imaging. So Google Health developed an AI system that has been shown to outperform radiologists in detecting breast cancer from mammograms in certain cases. So this system integrates into screening workflows, helping radiologists identify early signs of cancer more accurately, and this can significantly improve early diagnosis and patient outcomes. Okay, next up we have fuzzy logic. Fuzzy logic is about reasoning in situations that aren't black and white. Instead of working with strict yes or no answers, it deals with degrees of truth, which is a more flexible way to think about problems. This is especially useful for systems where there's lots of uncertainty or where precise answers just aren't realistic. So in bioinformatics, fuzzy logic has proven valuable in gene expression analysis, where the relationships between genes are often complex and not fully understood. For example, tools like fuzzy gene regulatory networks, or FGRN, use fuzzy logic to model how genes interact. So instead of saying a gene influences another in an absolute way, it maps these relationships in terms of probabilities or partial truths. This makes it easier to predict how genes impact each other in nuanced ways. And then finally here, we have knowledge representation. Knowledge representation is about structuring information so machines can reason, make decisions, and solve problems. While it isn't AI in the sense that it learns or independently performs tasks, it provides a well-organized framework of data that other AI tools can access and use. For example, in bioinformatics, a good example is ontology-based tools, which are basically standards for naming conventions and organizing data. One such tool is the Gene Ontology Database, which categorizes biological data into hierarchies, describing gene functions, processes, cellular locations, and more. This structured representation helps researchers answer questions like what genes are involved in a specific biological pathway, or which processes are active in a disease state. But one reason these knowledge representations are particularly powerful is because you can integrate them with other tools. For example, natural language processing could be used alongside the gene ontology to extract insights or even update databases automatically with new findings. Because these systems are built on structured frameworks, they'll be essential as AI becomes more embedded in bioinformatics workflows, helping not only with storing biological data, but also with tracking the models, parameters, and decisions made by AI during analysis. This level of structured logging will allow for more robust and reproducible science. We'll explore that future more in the next video, but for now that wraps up our look at specific AI models. Hopefully this gave you a solid overview of how AI is currently shaping bioinformatics and genomics without getting too deep into the weeds. Now for the rest of this first video in this three-part series, let's shift our focus to two of the most pressing challenges facing AI in bioinformatics and genomics today, and they are data and compute. Okay, first up, the data bottleneck. This is not a new problem. All these incredible breakthroughs, from classifying cell types to predicting protein structures, depend on one thing: high quality data. And in bioinformatics, that data is often noisy, incomplete, and biased. As I said, this problem isn't new, and it's become even more important now with the rapid advancements we're seeing with AI. So what are the solutions to this? Well, I think there are two major approaches. First, collect more /better/different data and second, make better or different use of existing data. I actually came across this article on our posting from 𝗕𝗶ecie𝗵𝗁, which is a good read for those of you who want to look into this further. But essentially he argues that the next breakthrough for machine learning in biology won't come from better algorithms, but from better or rather more diverse data. And this will actually come through wet lab innovation rather than further improvements in machine learning models themselves. The reasoning is that some biological datasets, like protein sequences and structures, have reached a point where more of the same data yields diminishing returns. This is because more data only improves performance if it adds new information. Otherwise, it can introduce redundancy or bias. Future breakthroughs will rely on new, diverse data modalities, not just more quantity. That said, it also depends on what you're studying. There's still plenty of areas where scaling existing datasets would be beneficial. Take genome sequencing databases, for instance. These databases are heavily skewed towards populations of European descent, comprising around 80% of sequenced genomes. This is because early genome sequencing efforts were concentrated in regions like Europe and North America, where funding, technology, and infrastructure were more readily available. This bias means that predictions for disease outcomes, drug responses, and genetic risk factors often perform well for individuals of European descent, but fail to account for critical nuances in underrepresented populations. But in general, yes, collecting new types of data, whether that's single cell measurements, spatial transcriptomics, wearable sensor data, or anything else, could be a real game changer. These will allow current machine learning models to capture layers of biology that traditional bulk datasets miss, potentially uncovering new patterns and insights. Now to the second solution for the data bottleneck, and that is to optimize utilization of existing data. Now, one idea that springs to mind here is multimodality, which is the idea of combining datasets from different sources or modalities. So for example, if we could say bring together gene expression data, protein sequence information, clinical information, pathological imaging data, among others, we may be able to uncover insights that might otherwise stay hidden when we use any one of these modalities individually. Deep learning models are particularly well suited for multimodal data because they excel at finding complex patterns and relationships that might not be immediately apparent. This is an exciting frontier that could drive significant advances in both AI capabilities and scientific discovery. So yeah, the path forward isn't just about collecting more data, it's about collecting better data, integrating it intelligently, and making sure it represents the diversity and complexity of the real world. Alright, let's talk about another glaring issue in AI for bioinformatics and genomics, and that is compute. So to power AI, we need computers, but not just any computers. These days it's all about GPUs. GPUs are the workhorses behind deep learning and even some traditional machine learning methods. They're designed for parallel processing, which makes them perfect for handling the massive datasets and complex calculations that AI thrives on. But here lies a problem. GPUs generally use more energy and power compared to CPUs or other processing units. And with the growing demand for AI, the need for GPUs is skyrocketing. Now this brings us to an interesting question. How do we deal with this increasing demand for compute? One possible solution is better software or better algorithms. Algorithms which don't just rely on throwing more GPUs at the problem. A great example of this is DeepSeek v3, which was released in December 2024. DeepSeek managed to rival the performance of OpenAI's GPT-4o in several benchmarks, particularly in coding and math tasks, despite only using 1/10 of the GPU hours needed for training, and at a fraction of the cost. If you're wondering what GPU hours are, they're essentially a way to measure the amount of computational work done by GPUs over time. So 1 GPU hour means running 1 GPU at full capacity for 1 hour, etc. Now DeepSeek was able to achieve this efficiency through algorithm optimization, which is a fascinating development that could reshape how we think about scaling AI models. And if you consider the environmental impact of big data centers, those massive facilities that house thousands of GPUs, this kind of efficiency is not just good for AI, but it's good for the planet. Now let's talk about genomics specifically. If you watched my video from a few weeks ago, you saw how file compression algorithms in genomics evolved over time, becoming specifically tailored to the unique characteristics of genomic data. Now I predict we'll see similar specialized developments for AI training and compute usage in bioinformatics and genomics, essentially creating DeepSeek-like efficiency gains, but optimized for the distinctive features of genomic data. Future algorithms will likely leverage the inherent patterns and structures found in genomic sequences, such as repetitive elements, use conserved regions and biological motifs to dramatically accelerate processing while reducing computational requirements. Just as specialized genomic compression algorithms outperformed general-purpose tools, these genomics-optimized AI approaches could potentially achieve orders of magnitude improvements in efficiency compared to general AI models when applied to biological data. Okay, let's go on to the second solution now for the compute problem, which is better hardware. So right now, most AI models are trained on general-purpose GPUs, originally built for graphics. But that's starting to shift. We're seeing the rise of AI-specific chips designed from the ground up for machine learning workloads. One example is TPUs, or tensor processing units, which are purpose-built for AI. This on screen is Google's new Ironwood TPU, announced just this month. It's optimized for inference, meaning it's built to run trained models quickly and efficiently. Chips like these can offer major gains in both speed and energy use, especially for certain types of tasks. And it's not just about speed. Smarter hardware could also bring down costs, making cutting-edge AI tools more accessible to more people. This matters because the bottleneck isn't just intellectual, it's infrastructural. Plenty of people have great ideas but lack access to the compute they need to test them. So if we can improve compute efficiency, whether through better software or better hardware, we'll make AI more accessible, more sustainable, and better suited to the unique challenges of bioinformatics and genomics. Right, that is a wrap on where we currently stand with AI in bioinformatics and genomic sciences. We've covered the major AI approaches being used right now, from classic machine learning to cutting-edge deep learning, and how they're being used across everything from drug discovery to protein structure prediction. We've also unpacked two of the biggest challenges shaping the field: the data bottleneck, holding back model performance, and the compute demands, making AI hard to scale. In part 2, we'll look ahead, projecting what the next 20 years might hold. We'll explore some emerging technologies, potential paradigm shifts, and make some bold predictions about where AI and genomics could be headed. Then in part 3, we'll bring it back to you, what all of this means for your career and how to position yourself to thrive in this new era of science and technology. I've added links to the full 3-part series both above and below. The upcoming videos will be added to the playlist as soon as they're published. If you found this video helpful, then do hit that like button and subscribe. It really helps more people find the channel. Thanks for watching part 1, and I'll see you in part 2.